All right, so let's keep talking about prokaryotes. This is the second part in the lecture. Remember, go back and make sure you can highlight those kind of unique features or special characteristics and traits about prokaryotes that we covered in the first lecture. Keep that list in hand and in mind as we move forward. Something I will say, prokaryotes are a field of incredible opportunity. Their phylogeny is constantly changing. Molecular systematics, DNA analysis, all of that is changing what we know about prokaryotes. This is what led to the split of prokaryotes into the bacteria and the archaea. It's based on molecular, not on phenotypic. So that was back in 1972. Carl Woese from the University of Illinois in Champaign-Urbana started proposing this. Took about 30 years, but we finally moved to this new system. That's the 3-domain system. Okay, always opportunity in this field if you're interested in it. So something I just like to mention. So with that being said, I'm going to show you some of the current stuff, but I'm not going to ask you guys to memorize this because it is a moving target. So why memorize something that's going to be changing in the near future? Almost guaranteed by the time you're done with your bachelor's degree, this will be revised again. So to me, it's not relevant to memorize this chart here when it's going to change or is under revision as we speak. But the general picture, this is how we've grouped things. You got domain eukarya, domain archaea, domain bacteria. Underneath each of those domains, you have those groups. How archaea and bacteria get reorganized still remains to be seen. But what I am more concerned about you guys knowing, again, is what are the key features of these things? How do we identify them? What are the pros and the cons? How do they impact our lives? Those are the things that I want you to take away from this course. You go to an upper level course, they're gonna drill you much more detail on this stuff. All right. Now, when we look at archaea, there's some traits that they share with bacteria, but other traits they share with eukaryotes. They are actually closer related to the eukaryotes, hence the branching point right here, common ancestor branching point that gave rise to eukarya and archaea, versus the branching point back here that would tie to bacteria and then the common ancestor. So that just rocked the world when molecular data showed us this and gave us, created this revision to the phylogenetic tree of life. So again, a lot of really interesting things happening here. Something to mention, there's lots to do with this knowledge here. The prokaryotes have a large spectrum of metabolic diversity. Some of them exhibit one type, others exhibit multiple types of metabolic diversity, how they gain their energy. But energy and carbon sources, how they use those can be kind of organized into 4 categories, or we call them the 4 major modes of nutrition: photoautotropy, chemoautotropy, photoheterotropy, and chemoheterotropy. So just to keep it very brief, if you're an autotroph, you make your own energy. Auto refers to self-producer. Heterotrophs are organisms that break down pre-formed energy. So somebody else built it, we take it and break it down and get the energy from it. So humans are heterotrophs. Cats and dogs and pretty much all animals would fall under heterotrophs. Producers, plants, bacteria, algae, they use photosynthesis. Some use chemical processes to make their own energy. So that's the big difference here, where the 4 major modes of nutrition we'll see within the prokaryotes, is they can do this a lot of different ways. This, again, is what makes these guys very, very adaptable. Now, if we're going to compare the bacteria versus the archaea, we kind of have 4 key areas that make them different. Their plasma membrane, their cell wall, how they replicate their DNA, and then how they express their genes and the genetics that they contain. So those are kind of the 4 fundamental features we look at when we're identifying, is this organism a bacteria? Is this organism a member of the archaea? So it's, to a certain extent, it's a checklist. If you have this, check, 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 check. Okay, you're a bacteria. Now, what type of bacteria? Well, here's another checklist. If you're an archaea, oh, okay, you're over here because you have these features. What type of archaea? Let's look at the additional checklist or the additional features. Right? So that's what I want to talk a little bit about. How do we identify the difference between bacteria, archaea, and then the big general picture, the eukaryotes? So to make your life easy, here's a table. Right out of the book. Try to make it simple and easy for everybody. So, all right. So bacteria. Right here. Archaea are right here. And then the eukarya in that column there. All right, so do they have a nuclear envelope? No. Bacteria and archaea do not have nuclear envelopes. They don't have a nucleus. They have that nucleoid region. So if you have a cell with a nucleus, eukarya. Do they have membrane enclosed organelles? No. Bacteria and archaea do not. If they have an organelle, eukarya. Now, let's start looking at the cell wall. So here's your bacterial cell. They will have a cell wall around it. So that red line is the membrane. So draw this out. My recommendation for those of you who are kinesthetic learners, visual learners, draw this out. Make it large so you can label it. It's a lot easier on a piece of paper than trying to do it on a computer screen. So here's your nucleoid region. Here's your cell membrane. Here's your cell wall. I'm just going to do CW. A little easier to label with that. But I would write out, if I were you, please write out cell wall. That way you don't look back and go, what does CW mean? All right, so cell wall. Bacteria have one. And they have peptidoglycan in their cell wall. Peptidoglycan is this little structural carbohydrate. Archaea have a cell wall, but they do not have peptidoglycan. So there's a lack of peptidoglycan in the cell wall of the archaea. Wall is there, does not have peptidoglycan. So if you could zoom in to a cell and look for peptidoglycan, if you see it, you know you're in bacteria. If it's not there, you're archaea, or you might also be eukarya, because there are cell walls in eukarya. So that's one of the features, that cell wall. When we look at the membrane, the actual cell membrane, there's certain components in the membrane. It's not listed in the entire membrane, but it's listed as membrane lipids. The membrane lipids in the archaea, they have a glycerol component. So if I'm looking at archaea, I'm going to put it up here. GLY is going to be my abbreviation, since I can't write really well with this. So glycerol is present in the archaea cell membrane. That's what makes them resistant to temperature. We don't have that. We can't handle high temperatures, neither can bacteria. But archaea can. Some of these suckers live in volcanoes, people. You have to have some physical structure that enables you to survive in that environment, and that's the glycerol component of the cell membrane. Adaptations. All right, so those are 2 of the key features. So I'm going to put a check mark or a little X. So cell wall, cell membrane. Okay, again, remember, sorry, did them backwards. Membrane, cell wall. Now DNA replication and gene expression. All right, so if you're a bacterial cell, and let me get back to my green. Actually, I'll stay in red. And you're a bacterial cell who's going to go to DNA replication. What you're going to do is you're going to replicate in a big circle. So I'm going to expand this out. Let's say here's the chromosome. That's the DNA. Okay. So what bacteria do is they start here with their DNA replication and then just go whoa, getting exhausting. Going through millions and millions of base pairs. And then they get to the end and they're, okay, I'm done. And now I have another big loop of DNA. Finished. All right, that's bacterial replication. Ooh, see if I can do this without. Oh, sorry. All right, let me go back. Here's our Archaea. There's their DNA. Now, Archaea do this a little bit differently. Give me a different color. They're going to go like this, and they're going to start in this direction. But while that's happening, they're going to start here and go in that direction and work towards that point. And then maybe they're going to go in this direction and work like this and start here and work. So get the point? Archaea have multiple replication points. Archaea DNA replication is very similar to what we do, where we have multiple points of replication occurring simultaneously. Boom. There's a big difference. This is one of the reasons why Archaea are closer to us than they are to the bacteria, is because how we replicate our DNA. All right, so the last major thing to mention is gene expression and how Archaea and bacteria will express their genes. So if you remember, when we look at a piece of DNA, this is a very, very poor drawing of a DNA strand. There are chunks of the DNA that get cut out and left within the nucleus when we do our DNA turning into RNA, turning into a protein expression. So remember DNA, that's supposed to be DNA, becomes RNA. But before the DNA becomes the RNA, that's DNA to RNA is that step we call transcription. Certain pieces get cut out. Those are known as the introns. Introns get cut out of the DNA and basically stay in the nucleus when we're talking about eukaryotes. We have introns in a lot of our genes here, in many of the genes. Archaea have them present in some of their genes. But when we look at bacteria, introns are very rare to see these things in the bacteria. Bacteria pretty much use their entire strand of DNA, convert it to RNA, convert it to a protein. So that creates a significant piece of information or difference between prokaryotes that split them into the bacteria and then the archaea. All right, so big main differences. The molecular classification is how we're identifying these guys now. They're using amino acid sequences for key proteins. What's your percentage of guanine to cytosine in this one versus that one? Nucleic acid hybridization. How can you base pair between different ones? The gene and RNA sequencing, what we were just looking at. They're especially looking at ribosomal RNA and then whole genome sequencing. So lots of different ways to classify. And again, guys, it's a moving target. Right now, this is what we're looking at. Same diagram we've been looking at and seeing. Common ancestor gives rise to domain bacteria. And then this line. This line then gives rise to archaea and eukarya. So archaea and eukarya have a closer ancestor than archaea and bacteria. Check out the next part for some Archaea features.